Good morning students. Welcome to Ligase Academy, your educational catalyst. In this video, we will be dealing with a very basic topic of molecular biology, that is the structure of chromatin and chromosomes. Whenever we study a topic on life science, we usually come across with this term called as DNA. So what is a DNA? Deoxyribose nucleic acid, right? If someone asks you what is a DNA, we usually say that it is the unit of inheritance and it is a double helical structure, etc. But do you think that this DNA is present as a double helical structure within our cells? No. We all know that every organism is made up of millions of cells and every cell consists of DNA. So how is this long thread-like helical DNA packed within the minute cells? That is what we are studying in this video. Before moving to the structure and packing of DNA, let's go through some basic terms. The first one is DNA. So what is a DNA? It is a molecule that carries the genetic information. So what is the difference between a DNA and a gene? Gene can be termed as the basic unit of heredity. Genes are the segments of DNA that codes for proteins. That is where we say every individual consists of different genes, and that is why every individual is unique in their appearance. Then what is chromatin? They are the substances present in nucleus that consist of DNA and proteins. So when there is cell division, these chromatin will condense to form a chromosome. So the chromosome can be termed as the thread-like structures which consist of a DNA molecule. And another term is chromatids. So these chromatids are the identical strands of a chromosomes. That is, usually the chromosome consists of two identical strands that are called as sister chromatids. In the interphase nucleus, the genetic material is existing as chromatin fibers. When visualized under an electron microscope, the interphase chromatin exhibited a structure that resembled a beaded string. The beaded string form of chromatin has a diameter of 10 nanometer. The 10 nanometer fiber represents the basic organization of chromatin. So the chromatin is made up of DNA and proteins. There are two types of proteins, that are histones and non-histones. And based on the relative proportion of arginines and lysines, that are the basic amino acids in histones, histones are divided into 5 classes: H1, H2A, H2B, H3 and H4. So they are called as the canonical histones. Of these 5 classes, H3 and H4 are highly conserved in evolution with similar amino acid sequences even in totally unrelated species. The 10 nanometer fiber is arranged in the form of a beaded string where each bead is designated as a nucleosome. So nucleosome can be considered as the basic unit of chromosome. These repeating nucleosomes make up a chromatin. So usually a nucleosome consists of a core particle, that is the histone octamer, and 2 turns of DNA. As we all know, DNA is negatively charged because of the presence of the phosphate group, whereas the histones are positively charged because of the presence of the basic amino acids, that is lysine and arginine. So we have already said that there are 5 different types of histones, and here the H3 and H4 histones will form a dimer, whereas the H2A and H2B form another dimer, and these will combine to form a tetramer and further an octamer. So the simplest form of chromatin is the beads on a string structure with DNA coiling around the histone octamer as the beads. The histone octamer can be considered as a flattened disc or a cylinder with a diameter of 11 nanometer and a height of 6 nanometer. So usually the DNA takes 2 rounds around the histone octamer. And the length of the core DNA is invariable in all species and all cells. And the role of histone H1 is to stabilize the 2 turns the DNA takes around the histone octamer. The adjacent nucleosomes in the 10 nanometer fiber are connected by a linker DNA. its length is usually variable but maybe 8 to 114 base pairs. So we can sum up that the beaded string structure can be set as the basic level of chromosome arrangement. So each bead can be considered as a nucleosome, and the DNA is taking 2 turns around a histone octamer, which is called as a nucleosome, and each nucleosome is linked by linker DNA. Thus, the repeating nucleosomes will make up a chromatin fiber. A 10 nanometer fiber then folds into a 30 nanometer fiber. The 30 nanometer fiber represents the second level of organization of chromatin. In interphase nucleus, both the 10 nanometer and 30 nanometer forms can be seen. So usually the 30 nanometer forms can be seen in two different forms, solenoid form or zigzag form. It is unclear which one of them is predominant in the cells. In case of solenoid form, one nucleosome will be interacting with the adjacent nucleosome, whereas in zigzag form, one nucleosome will be interacting with the alternate nucleosome. When the chromatin is stained, certain regions of the chromatins obtained dark stains, whereas other regions stained lightly. The darkly stained chromatin regions are called heterochromatin regions, which are transcriptionally inactive. The lightly stained regions are called euchromatin regions that are transcriptionally active. During cell division, the 30 nanometer chromatin fibers coil and condense to form metaphase chromosomes. So whenever we are talking about chromosomes, we are actually specifying the X-shaped metaphase chromosomes. A metaphase chromosome has two chromatids. The chromatids are the daughter chromosomes formed as a result of chromatin duplication during the S phase. In a metaphase chromosome, the sister chromatids are held together at the primary construction of the chromosome. Each sister chromatid is thinner at the primary construction. It is present in all chromosomes, whereas the secondary construction is not present in all chromosomes. The secondary construction is also known as nucleolar organizer region, since it is involved in the formation of nucleolus. And we can see a complex button-like structure at the centromere. It is usually assembled at the centromere during mitosis, which is called as kinetochore. Kinetochore of the chromosomes serve as the microtubule binding sites for the mitotic spindle. And at the end of the chromosome, we can see a region called as telomere, which is a region of repetitive DNA sequences. Each time a cell divides, the telomere becomes slightly shorter. So the main function of a telomere is to maintain the chromosomal stability and prevent the chromosomal degradation. To conclude, we can say that the DNA is existing as a double helical structure which will form to a 10 nanometer nucleosome structure, and that is the beaded string-like structure, which will form to a 30 nanometer solenoid or zigzag form, which will further condense again to a 300 nanometer, and again the condensation occur to form a 700 nanometer structure, and finally it will form a 1400 nanometer X-shaped metaphase chromosomal structure. So that is all about the basic level of organization of chromatin fibers and chromosomes. Stay tuned to our new videos on the basics and fundamental processes of life. Thank you for watching. Please like, share and subscribe Lifescience Academy, your educational catalyst.